histone, and their role in cell. We have seen that the nucleosome is a structure which is very important in tightly packaging of the DNA, so that the chromosomes can be in a very compact form, and whenever it is required, then these histone or these nucleosomes will be processed, and this winding unwinding will be there. So this is very important in the all kind of DNA activities, whether it is replication or whether it is a transcription. So in all cases the activity is important. So we have seen that these are the proteins. They are abundant in lysine and arginine. These two amino acids are too much, or they are highly present in that. They are the chief protein components of the chromatin. They are acting as a spools, or it is acting just like, in a reel, you call it as during your kite days, or even in thread. They are acting as a spools around which DNA is wound, and it is playing a role in the gene regulation. Without histone, the unwinding of DNA in chromosomes would be very long. As I told you, it will be length width ratio is more than 10 million to 1 in human DNA. So each human diploid cell, which is containing 23 pairs of the chromosome, has about 1.8 meters of DNA, which is wound around histones. And you can think about it, which has been put into very small micro size cells and its nucleus. So it is in a micron size. So a diploid cell, it has around 90 micrometers, that is 0.9 mm of chromatin. So which is when the diploid cell are duplicated and condensed, the result is around 120 micrometers of the chromosomes which are present. So this much structure, and if they are not properly wound, they will be entangled, they will not be properly expressed, processed, and therefore they are there. So thus these are the proteins. We have seen them. There are 4 basic type of histones: H1, H2A, H2B, H3, H4. Particularly these last 4 are important. H1 and H5 are not that prominent. So histone H2A, H2B, H3 and H4 are known as the core histones, while histone H1 and H5 are known as the linker histones. So they are forming the linker one, they are forming the linking it. They are present as a dimer. If you remember, we have seen this kind of figure earlier, and we have seen that they are present, how they are present in a various structure. So they are acting as a dimer, one and another one, they are mixed like that, and these dimer are having the activities. So they are similar in, they are all possess the histone full domain. 3 alpha helices means one is a helix, and more than one is, we call it a helices. They are linked up by 2 loops, and this helical structure that allows the interaction between the distinct dimers, head to tail fashion, or it is also called as a handshake motif. That handshake means just 2 hands. When you shake the hand, then both hands of same side, means right to right you cannot match, but right to left means your right and left, you will find that if the person is means right, your right and next person's left, you cannot match it, but when you handshake right to right, then you can match it properly. So this is how handshake motif is there, which makes the proper matching. The result of 4 distinct dimers then come together to form one octameric nucleosome core. We have seen nucleosome just now, just before this lecture, and it makes approximately 13 angstrom units in a diameter, and it causes winding of around 146 base pair, that is BP of a DNA, which is around the core particle. That is 1.65 times in a left-handed superhelical turn, to give a particle of around 100 angstrom units. The linker histone H1 binds the nucleosome at the entry and exit sites of the DNA, thus locking the DNA into a place and allowing the formation of higher order structure. So higher order means still we have seen more folds are there, and what we have seen it, we call it as a bits on a string structure. If you remember, we call it as a higher order structure. So this was earlier, we have seen the figure. The most basic structure formation is the 10 nanometer fiber of beads on a string conformation. It involves the wrapping of DNA around nucleosome, and it approximately 50 base pairs of DNAs are there which are separating the nucleosomes. Then higher order structures include 3 nanometer fiber, tail of that that is there, forming a irregular zigzag, and 100 nanometer fiber. And these structures are there during mitosis and meiosis. The condensation of chromosomes is there. They are assembled through this interaction. They are divided in variety of types, and they are expressed during the S phase, that is synthesis phase of the cell cycle. The replication independent histone variants are also present. They are expressed during the whole cell cycle. In animals, the genes which are encoding the canonical histones, they are typically clustered along with the chromosomes, and see, they are always required, so they are present near that, and immediately these are proteins are formed, and they lack any kind of introns because immediately it has to be functional, so there is no time for splicing, etc. They are usually, they have introns, and their messenger RNAs are regulated with the poly tails. So these are the complex multicellular organisms. They have a higher number of histone variants and variety of different functions. So these are the various families. Actually, we have seen main is as a H1. So these are the families, subfamilies, and the members of the family. You need not to remember each and every of that, but you can see the linker. We have seen H1 is a linker one. Here it has got subfamily of H1F, H1, H1, and in that you will find that various kind of around in H1F there are 4 members in the family. Then in H2A, H2B, H3 and H4, they are the main core histones. In that also we have in H2A there are 3 important subfamilies, that is H2AF, H2A1, H2A2. Here in B you will find F, A1, A2, B1, B2. In H3, A1, A2, A3. In H4 you will find H1, H41 and H44. So these are different subfamilies are there, and they are having so many structure, and these structures are responsible for a different kind of functional activities. Okay, so these are the structures which are there, and as we have seen earlier, they are making dimers. H2A, H2B are dimer, and H31 and H4, okay, they are dimer, and together H12A, H2B, H12A, H2B, H3 and H4, they are total 4 are causing a tetramer. So they are causing nearly 2 symmetrical halves. So we have seen these are the symmetrical half are there. In this figure we can see this, and these are the halves which they are by tertiary structure, and in such a way that the one micromolecule is the mirror image of another one. So like that half structure will be there. This is 1/4 quarterly, one after another. These are the dimer which are there. So this dimer has formed into a tetramer, okay, and these 2 tetramer will form the octamer. So this final structure will be a round shape which will be an aptamer. So these are the where the helix turn helix will be there, that is DNA binding protein that will recognize the specific DNA sequence, and accordingly the process will be there. Then they also share the feature of long tails at one end, and the amino acid structure, it is location of post-translational modification. We shall be seeing post-translational modification as a separate topic later. Okay. So what are the interaction it is having? Actually 5 types of interaction all the histones they are making. Helix dipoles, they form alpha helices in the H2B, H3, H4, and that causes a net positive charge that will cause accumulation of this point of interaction. and along with the negatively charged phosphate groups on the DNA. So this H2B, H3 and H4, they are having positive charge, while the phosphate molecules on the DNA will have negative charge, and because of that the positive negative interaction will be there. Then hydrogen bonds between DNA backbone and the amide group which are present in the main chain of histone proteins, so they will form hydrogen bonds. Then non-polar interaction will be there between histone and deoxyribose sugar. Then it will form salt bridge, that is normally Na plus Cl minus, like that. So they will form salt bridges and hydrogen bonds between side chains of basic amino acids, particularly lysine and the arginine, and also phosphate oxygens on the DNA. Then non-specific minor groove insertion of H3 and H2B terminal tails into two minor groove each on the DNA molecule. So these are important 5 places where there is a DNA will have interaction. You can note that there is no covalent bonding. It is all the weak bonds are there which can be replaced, which can be modified, or some higher energy can be converting or making this bond to break, because covalent needs very high energy. So these are not covalent bonds, these are the small simple bonds. They are having enough strength only to hold it together for some time, and because of that it maintains the dynamic structure. The highly basic nature of histones, apart from that, it contributes to their water solubility. They are subject to a post-translational modification by enzymes. Post-translation means after translation, after protein synthesis, whatsoever modification takes place, we will call it as a post-translational modification. Primarily on their N-terminal tails, but also in their globular domains. And such modification increase methylation, citrullynation, acetylation, phosphorylation, sumoylation, ubiquitination, and ADP ribosylation. These are the important, and because of this modification, because of this, it causes the changes in the function of gene regulation. So one of these activities, acetylation or deacetylation, we shall be seeing later, but then we have to understand that these all various activities are important. So just simple chemical modification on the histone or enzymatical action on the histone, like this methylation and all. So the term is simple. Methylation means methyl group is attached, citrullination means citrulline group is attached, acetylation means acetyl group is attached, phosphoryl group. So these are various group, ubiquitin group, ADP ribo, or this sumo, this group, they are there, they are attached, and accordingly these groups, once they are attached, because of that the structural modification is there, and that will affect the bonding, that will change the gene regulation or gene expression. So in general, genes are active, which are active, they are less bound histone, while inactive genes are highly associated with the histone during interphase. Okay. Also appear that the structure of histone has been evolutionarily conserved, and any deleterious mutations would severely maladaptive. All histones have highly positively charged N-terminus with lysine and arginine. So these are the history. I will not go for history. I will not go for conservation across the species. What are the function of this is important. It will cause compacting of the DNA strands. We have seen how they are doing, how these structures are playing important role. We have seen all this already. This is monomer, 1 plus 1 becomes dimer, this 4 becomes a tetramer, and all this 8 becomes an octamer. So this is what we have seen. This is many times we have seen this during nucleosome also. Okay. So these are how these structures are there and how they are arranged in the sequence. So basically these are 4, and that is H2A, H2B, H3 and H4. So we have got different colors of this, and you can see how they are arranged. Okay. So H2B is red, H2A is yellow, H3 is blue, and H4 is green. So see, these are the how they are there, and they are like a mirror image. So if it is top, then it will be at bottom. Red was at top, so now red will go at the bottom, and accordingly these 4 they have come together. and thus the dimer and octamers are there. Okay. So what are the functions of these? They are compacting of that. It causes very important compaction necessary to fit the large genomes of the eukaryotes inside the cell nucleus, and it is compacted 4000 times shorter than the unpacked molecule. So that much compression it makes. Then it is important in the chromatin regulation. They undergo post-translational modification that alter their interaction with the DNA and nuclear proteins. The H3 and H4 histones have long tails protruding from their nucleosomes, and these tails which are there, they can be covalently modified at several places, and this modification of this tail causes, we have seen all these processes, methylation, acetylation, etc. And these processes, because of this covalent bonding, there will be changes, and because of the changes there will be functional changes. We shall see what are the impacts of this. The core of histone H2A and H2B can also be modified, and the combination of modification are important in that. They are also called as a histone code, because there is a code which is present in DNA itself, and they will code as a histone code, and accordingly the modification will be there. Histone modification are very important in the processes like gene regulation, DNA repair, and chromosome condensation, mitosis, spermatogenesis, etc. Okay, in all cases it will be important. Then the next thing is, just a minute. So they are in how the histones are named, the nomenclature that includes few things. The name of histone, that is H3. Then amino acid, which is important in that, that is single letter of amino acid, that is K for lysine. So we shall see their structures just after this. So single letter amino acid abbreviation, so K for lysine is there. Then type of modification, if it is methylated covalently bonding, or we call it as a post translation modification. So if the modification is methylation, we will call it as a ME. If it is phosphorylation, we will call it as phosphate. If it is acetylation, we will call it as AC. If it is ubiquitin molecule is attached, or ubiquitination is there, then we will call it as a UB. Then how much number of modification are done on that? Okay. So particularly in ME, that is methylation. Okay. So here ME means not me. Okay, me means I, me, what we call it, is not like this. So here methylation. So only methylation is known to occur in more than one copy per residue. So it is 1, 2 or 3, that means monomethylation, dimethylation, trimethylation, like that. So example of this now, how histamine we will be naming. We have seen various families and superfamilies. So we have seen subfamilies and families. So H3K, K means lysine. So in the H3 there is a lysine is important. At what position? 4 position. Okay. Then Me, that is methylation has done, and it is what? At only one methylation. So it is a monomethylation of the 4th residue of lysine. Okay. That is a lysine from the start. That is from the N terminal of the H3 protein. So this is how the nomenclature is made. So nomenclature is made in this sequence. I am not expecting you to remember all the nomenclature and all the function, but yes, you should be aware of it. So these are the families and the various names of that. So okay, type of modification: monomethylation, dimethylation, trimethylation, acetylation. Okay, like that. Now here K means what? With this histone, lysine is there. At what? 4. So what is the job of that? It will cause activation. Okay. But some of this example is where there is a dimethylation and H3K, that is at 3 position, lysine 2,7. So that is causing reparation. So these are the reparation. So they are reparation. Or here H3K9 is causing reparation. Reparation means stopping of the expression, gene expression. So we call it as a repression. So either it will cause activation or repression. So these are the various activities which are there. These are the histone modification, or we call it as a post-translational modification in the transcription and regulation. So what are the functions of the histone modification? Okay, so these are the 4 basic histone, isn't it? H2A, H2B, H3 and H4. So there we can have any molecules that can be there. Example, methylation, acetylation, then citronylation, phosphorylation, ubiquitination, etc. So there you can find that on amino in the sequence you can find that the sequence in that you will have this various. So here K means what? Lysine, we have seen. Okay. So various amino acids are there, and as per these amino acids, the names will be there. Okay. So these are the amino acids, accordingly names are mentioned. The huge catalog of histone modification have been described, but functional understanding of most is still not there. Only few we are knowing that what will happen because of that. It is thought that histone modification may underlie a histone code, and therefore this combination of histone modification has specific meaning. Some of the meaning are understood, some are not understood, or rather many are not understood. So most functional data concern individually prominent histone modifications that are biochemically amenable to detailed study. So these are various modifications. Now you understand from this structure that various amino acids are there on the what we can say tail positions of each. So these are the tails of all. In tails there are the amino acid sequence. On this amino acid sequence, wherever there is any group is attaching, accordingly we are making the name. I am repeating this what just now we have seen about nomenclature. I hope you can see the figure. So this is the basic histone structure. These are various histone families. Okay. So here is H2 family, H2B family. In that various amino acids are attached. That is, the tails are there, and in these tails there are several amino acids are attached. On tail, if this is a lysine K, means wherever K is there, either it is acetylated, methylated. Okay. So these are various amino acids, and these amino acids are accordingly modified. Okay. So the most prominent one is the lysine, and therefore the example is given of lysine, but there are other also. So here see, we can see lysine methylation is there. Then so whenever lysine is having any chemical, this one, so it will be methyl lysine, dimethyl lysine, trimethyl lysines, like that. So it will be there because of the attachment of the methyl group. Then next one are the glutamine, arginine. With glutamine, serotonin molecule is attached. The serotonin group in the position of 5-glutamate of H3. It particularly happens in the serotonergic cells and neurons, and this is the part of differentiation of the serotonergic cells. And this post-translation modification happens in along with this H3K4Me3 modification, and it potential binding the transcription factor. These are various transcription factors. So because of that, it is becoming fast. Okay. So then arginine methylation is another activity. Then arginine citrullynation. So at arginine group, citrulline is attached. Lysine acetylation. So on the lysine again acetyl group is attached. Then serine, threonine, tyrosine, phosphorus. If you remember, in apoptosis, these serine, threonine play important role. Tyrosine kinases, they play important role. So these are the tyrosine kinases are there, and accordingly they are all functions in different kind of transcription control. So example of these are which are actively transcribed in the gene. Very common modification are associated with the active transcription, that is a trimethylation of H3 lysine and trimethylation of H3 lysine 3,6 and at 4. Okay, so H3K for Me3 and H3K 36 Me3, these are the important one which are there, and there are various repressor gene also there. Some are activated, means they are activating the gene expression, while there are many which are the repressor gene. Okay, so you just understand various repressors and activator, that they are there, they are making many function. Apart from that, they are promoters, means they causes initiation of the replication or transcription, so we call them as a promoter, from where it will start the actions. Okay, and they are important for cell differentiation, etc. So these modification mark genes that are ready for transcription. They are not required in a stem cell, but they are rapidly required after differentiation into some lineages. So once the cells start to differentiate, these bivalent promoters are resolved to either active or repressive state depending on the chosen lineage. Then what are the other functions they are doing? What are the important function of these various histones? They are marking they are important in the DNA damage. So they are mark the sites of DNA damage, and because of that it can be repaired. It also protect DNA from the getting destroyed from the ultraviolet radiation of the sun. Okay, then example of these are the phosphorylation of H2AX at a serine at 139 and acetylation H3 lysine at 56. So these are the there. Then it is playing important role in the DNA repair. Example of this is a trimethylation of H3 lysine 36, so H3K 36 Me. You might have seen this is very prominent one at almost many places we have seen this. Then next, and even this H3K 36 and 56, they are quite important. Overall lysine is important into that. Then they are, as I told you, DNA repair. Then next is they are important in the chromosome condensation. Then some of these are found in the addiction. They are responsible for addiction. You know about addiction or drug abuse we have seen earlier, or drug dependence if you remember. So like nicotine and many other substances are there, alcohol and many such substances there which are causing addiction. So these are the important epigenetic modification of the histone tail in some of the region of the brain. They are found to be related with this kind of addictions. So then I will not go into synthesis of this because we know how it is there, and this is how histones are playing important role. So what we have seen today is what are various histones, what are their various structural and functional features. Okay, how they are nomenclature, what kind of tails are there, and what post-translational changes, that is after formation of histone everything is there, what kind of sequences are attached, what kind of chemical moieties are attached, like whether it is methylation, ubiquitination, acetylation, etc., phosphorylation. So what happens to that, and because of this what kind of functions they are playing, that we have seen. So in brief, histones are important for nucleosome formation. Nucleosome is important step in the compaction of the long DNAs into very small chromosomes, and not only just haphazard condensation or compression, but it is in such a way that whenever whatsoever part is required, that can be expressed, so that at that particular time that protein synthesis or replication or whatsoever it is there, that should come out, and this is what importance this is making. So therefore histone is very important. There is always link between cell cycle control and machinery and histamine synthesis, that during synthesis phase these are histones are synthesized, and that is S phase, and they are playing very important role into this. So this is how the histones are there, and they are playing important roles. So I think this much is enough for us. Next portion we shall see how the histones are acetyl transperases are there, deacetylation is there, and how they are important. Okay, so there we have seen various kind of histones. So take care. If you have any difficulty, you can ask. Okay.